Hello again. Today we are going to be looking at another important topic again. But before we go into that topic, I would like to ask you this wonderful question. Whose genes are more dominant, mother or father? Whose genes are more dominant? Is it the mother or is it the father? I know this will be a kind of a tussle between parents, but the actual fact is that it is actually the father's gene that is more dominant. I'm not trying to be a gender racist, but it is more of the father. Check statistics or records have also shown, for instance, if you have a tall parent or a tall male get married to a tall or a short female, you will discover if they have about 3 children, you will discover that out of those 3 children, you will find out that more of the father's traits are being expressed, which are dominant traits, are being expressed in those offspring more than that of their mother, okay? So we find out that the father's gene are more dominant compared to the mother's gene. So today we're going to be looking at the topic genetics part 2, but in this particular topic we will be narrowing our discussion to one of the, will I call them, great men that actually made so much discovery about genetics, and we will be looking at Mendel's work or experiment in genetics, okay? Now, the subtopics we're going to be looking at in this particular topic is who is Gregor Mendel. We're going to explore a little about himself or his life, though he's late. We're going to talk a little about Gregor Mendel, very, very brief. And then we'll look at his experiments. We are also going to be talking about 2 important methods of his experiments, which are the monohybrid inheritance and dihybrid inheritance. Okay, now if you are set, let's take a look at the specific objective. Now, one of the specific objectives is that by the end of this topic, learners should be able to mention reasons why Mendel used the garden pea plants. And number 2, you should be also able to explain briefly monohybrid inheritance and state the law of segregation of genes. And then finally, you should also, by the end of this class, be able to explain briefly dihybrid inheritance and also state the law of independent assortment of gene. The law of independent assortment of gene. Now let's begin with a look at who Gregor Mendel is. Now, Gregor Mendel was born in 1822 and actually he passed on in the year 1884. Now, Gregor Mendel, as you can see on the screen, was a monk in an Augustinian monastery in Austria. So he was a monk in Austria. Now, he is regarded as the father of genetics because his work formed the foundation for the study of genetics. His work formed the foundation for the study of heredity and also of variation. So this man is regarded as the father of genetics up till today. I have seen in several wire questions, even jam questions of this nature have been asked. Who is the father of genetics? Please take note, the father of genetics is Gregor Mendel, and he was awarded the father of genetics because his work actually formed the foundation for the study of genetics. Now quickly, let's take a look at Mendel's work. What was he working about? Now, Mendel's work or Mendel's experiment was focused on understanding how traits are being transmitted from one generation to another. He was trying to understand how these traits are being transmitted. Remember, we call that heredity. So how is it transmitted from one parent organism to another, which is the offspring? Now, Mendel worked with, what he used in actually working was not humans, it wasn't even animals, but actually the first thing he worked with was garden pea plants. And the botanical name for garden pea plant is Pisum sativum. Pisum sativum. Now, why garden pea plants? Why did he make use of garden pea plants? Now, Gregor Mendel actually decided to work with garden pea plants because of the following reasons. Number one, he discovered that garden pea plants, they can be self-pollinated, not apart from undergoing cross-pollination, they can also self-pollinate. And you're going to see some of the reasons why while he was carrying out his experiment. Number two reason why he chose the garden pea plant was because of their short lifespan. The garden pea plant has a very short lifespan. And in fact, garden pea plants are actually annual plants. They are not biennial, they are not perennial. In fact, they complete their life cycle within 1 year. Another thing, again, number three reason why he picked or chose the garden pea plant was that the garden pea plant showed several unique characteristics. And these characteristics, they occur in contrasting pairs. Well, what do we mean by they occur in contrasting pairs? I'll give you an example. He discovered that the garden pea plants, we have the short garden pea plant and we have a tall garden pea plant. They are contrasting and they are in pairs. We have short, we have tall. He discovered that also some garden pea plant seeds were round, while there are some seeds that are wrinkled. He discovered some seeds were yellow in color, while some seeds were green in color. He discovered that in garden pea plants, some of the flowers were red, and he discovered in some of them, their flowers were white. He also discovered in the garden pea plants, the pods. Some of the pods were smooth, and he also discovered that some of the pods were constricted. He also discovered that some of the pods in terms of colors were green, and he discovered some other pods that were yellow. Now, based on these several unique characteristics which exist or occur in contrasting pairs, Gregor Mendel actually discovered that they were perfect in his work or to carry out his experiment. Now, let's take a look at some of Mendel's experiments. Now, Mendel's experiment or work was based on what we call complete dominance. Complete dominance. In future or in our subsequent future presentations or lectures or topics, I'm going to be actually differentiating between complete dominance as well as incomplete dominance. Okay, there is also what we call incomplete dominance. Now, what exactly is complete dominance? Complete dominance is the ability of two contrasting characters or two contrasting pairs of genes to cross and produce what we call a heterozygous phenotype that is the same with the contrasting allele. I'm going to repeat it. I said it is the ability of two contrasting alleles to cross and produce a heterozygous phenotype that is the same with the two contrasting allele. And we're going to see examples of this. Let me give you an instance. Now, I have a tall plant and I have a short plant. Now, both of them, when they cross, they give rise to a heterozygous. Remember, the pure tall plant is homozygous, big T, big T. The pure short plant is homozygous, which is also small t. But when they cross, they give rise to big T, small t, which is heterozygous, but still carries the same phenotype as one of their parent organism. That is, the parent organism that has big T is tall. The offspring, which is big T, small t, is also tall in terms of the phenotype. But in terms of genotype, they are what? Contrasting. The genotype of the parent organism is homozygous, while the genotype of the offspring is heterozygous. Now, Mendel employed two methods in conducting his experiments. The first one he did was what we call monohybrid inheritance, and the second one is called dihybrid inheritance. Now, quickly, let's take a look at monohybrid inheritance. Now, what is monohybrid inheritance? Monohybrid inheritance involves a cross between two different plants with one pair of contrasting character. With one pair of contrasting character. Now, in this monohybrid inheritance, Gregor Mendel worked with a tall plant and a short plant. That is one pair. But the characters are actually contrasting, tall and short. Okay? Now, how did he carry out this experiment? Now, first of all, he artificially pollinated or crossed a pure tall plant with a pure short plant. Remember when I told you crossing simply means they breed, okay? Biological intimacy took place, or sexual reproduction actually took place. So he artificially crossed a pure tall plant, which is capital letter T, which is homozygous for tallness, with a pure short plant with a small letter t. Now, the next thing he did is he collected the resulting seeds from the cross and then planted it. Because after the cross, they will give rise to a seed. Now, this seed must be planted for it to germinate into a new plant. So he collected the resulting seeds and then planted it. When he planted it, he discovered that the resulting offsprings were all tall plants. There was no short plants. They were all tall plants. And he called these the F1 generation. He called it the F1 generation. But I'm going to tell you what subsequently he discovered. He discovered, in fact, this was what brought about the discovery of dominant and recessive genes or traits. Okay? So he came to the discovery of dominant genes and recessive genes. Now, after getting the F1 generation, he self-pollinated the F1 generation. That is, he self-pollinated it artificially, and the resulting seeds he planted again. Now, when he planted it, as those seeds started germinating and bringing forth plants, he discovered that the resulting offsprings or the resulting plants, some were tall and some were short. Some were tall and some were short in the ratio of 3:1. That is the ratio of 3 out of whatever resulted or whatever offspring came out, 3 out of the whole, okay, were tall, and 1 ratio out of the whole were short. Okay, now these were what he called the F2 generation. The F2 generation. Now this is what you're going to see in the cross. Now you can see the big T as tall plants and the small t as the dwarf plant, or you can call it the short plant. Now when they crossed, they produced a tall plant. They said all tall with the genotype big T, small t, which is heterozygous tallness. Okay, and these were the F1 generation. Now this F1 generation also underwent crossing. So he self-pollinated it and then got a result of which he had 3 tall plants and then 1 short plant, which I said in the ratio of 3:1. As you can see in the phenotype, we have 3 tall plants. In the phenotype, there are 3 tall plants, and in the phenotype, we have 1 short plant. So the phenotypic ratio is 3:1. But the genotypic ratio is 1:2:3. Why is it 1:2:3? We have 1 homozygous dominant tall plant. We have 2 heterozygous dominant tall plants. And then we have 1 homozygous recessive short plant. And that's why we called it, the genotypic ratio is 1:2:1. But the phenotypic ratio is 3:1. Phenotypic ratio 3:1, because the big T, small t, they are representations of tall plants, while the small t is for what? Short plants. Now, based on this discovery, Mendel proposed his first law of inheritance. And this first law of inheritance is called the law of segregation of genes. Law of segregation of genes. And this law states that genes are actually responsible for the development of the individual, and that they are independently transmitted from one generation to another without undergoing any alteration. Of course, you can see it. They are being transmitted from one generation to another, and these genes are actually definitely responsible for the development of characters in their offspring. So this is what the first law of inheritance proposed or propounded by Gregor Mendel himself, which he called the law of segregation of genes. Remember, I said that he carried out two methods of experiments. The first one he called monohybrid inheritance, and then the second one is called dihybrid inheritance. Now, what is exactly dihybrid inheritance? Dihybrid inheritance involves a cross between two different plants with two pairs of contrasting characters. A cross between two different plants with two pairs of contrasting characters. In monohybrid, it is two different plants with one pair of contrasting characters. Now, what are the two pairs of contrasting characters he used in dihybrid? He used seed shape and seed color. Seed shape and seed color. For seed shape, he used round and wrinkled, and then for seed color, he used yellow and green. Yellow and green for seed color. Now, this is how he carried out the experiment. He followed the same steps he followed in monohybrid inheritance. First of all, he artificially crossed plants with round and yellow seeds with plants with wrinkled and green seeds. Now, after crossing these two different plants that have two pairs of contrasting characters, he collected the resulting seeds from the cross and planted it. When he planted it, he discovered that the resulting offsprings were all round and yellow seeds. That was also a proof of complete what? Dominance. A proof of complete dominance. They were all round and yellow, and he called this the first generation. That's the first filial generation. He also self-pollinated that first filial generation, so that is the round and yellow seeds. He self-pollinated them and collected the resulting seed and planted it. Now, he discovered, when he planted the resulting seed, he discovered 4 phenotypes. In the other one, he discovered 2 phenotypes. In this one, he discovered 4 phenotypes in the ratio 9:3:3:1. 9:3:3:1. And he called this the second filial generation. Now, this is the structure that shows the cross. The first cross, which he did, you can see that the round and yellow seeds, and then this one is the wrinkled and green seeds. But when they crossed, they gave rise to round and yellow phenotypically. Now, when he now self-pollinated these round and heterozygous round and yellow seed plants, he discovered 4 phenotypes. Now, these 4 phenotypes are in the ratio 9:3:1. 9:3:1. Now, what is the phenotypic ratio 9:3:3:1? Now, the 9 stands for round and yellow seed plants. So it means out of all the 16 plants that actually came out as a result, 9 of it were round and yellow seed plants. 3 of them were round and green seed plants. 3 of them were wrinkled and yellow seed plants, and then 1 was wrinkled and green seed plants. Out of the 16, he discovered that 9 were round and yellow seed plants. That's the phenotypic ratio. 3 was round and green seed plants. 3 was also round and yellow seed plants, and then 1 was wrinkled and green seed plants. And then he now did also a genotypic ratio, which is 4:5. 4:5. He discovered that 4 of the resulting offsprings were homozygous plants. 4 homozygous plants, and he also discovered 5 heterozygous plants. 4 homozygous plants and 5 heterozygous plants. So this was his discovery. Now, based on this experiment he carried out, he came up with a law, which is the second law of inheritance. And this law states, or can also be referred to as the law of independent assortment of genes. The law of independent assortment of genes. This law states that each character behaves as a separate unit and is inherited independently of any other character. I repeat again, it states that each character behaves as a separate unit and is inherited independently of any other character. So that is the law of independent assortment of genes. Now, but before we go, and we've come to the concluding part of this particular topic, but before we go, please let's take a look at some few questions using the ExamGuide app. Very briefly, we won't take much time for this. Now, number 1 says, in the F1 generation, we've just talked about it, F1 generation of Mendel's experiment obtained by crossing pea plants of long stems with those of short stems. What was his observation from F1 generation? F1 generation, that was under monohybrid inheritance. The F1 generation, we discovered that 3/4 were long, and 1/4 was short. That was in the F2 generation. The correct answer is C. All were long. In the F1 generation, all were long. It was in the second generation we have 3:1, which is 3/4 were long, while 1/4 was what? Short. Then we have the next question. Look at this. These are animals. They said, what are the genotype of the parent if the gene for skin color is H? What are the genotype of the parents if the skin color is H? The gene for skin color is H. Now, spotted leopard, is it H, small h? And then black panther, is it small h? We have spotted as small h, big H, small h, and this one. So the correct answer is D, the spotted, because if you look at this cross here, you will discover that the spotted, they had the dominant traits. They have more of them. In the F1 generation, the resulting offsprings were all spotted. And in the F2 generation, the spotted was 3:1, which was what? The black panthers. So the correct arrangement, or the correct gene for both of them, both leopards animals, is that the spotted has capital letter H, which is homozygous dominant traits or gene, while the black panther has a homozygous recessive traits or genes. All right. And then finally, if a heterozygous red flower plant, big arrow, small arrow, was self-fertilized, the offspring will be expected to be what? The correct answer is B, which is 3:4 red flower and 1:4 flowered plants. As you can also see, we still have more questions under this particular topic on genetics. Thank you for participating in today's class. You can practice more questions using your ExamGuide app. The app scores and gives a detailed explanation of all the questions at the end of your practice test. You can also learn particular topics of interest with different modes, like study mode, mock mode, and even practice mode. It also has other features that makes learning very fun. Now, it is a must for all serious students. Download from www.examguide.com if you don't have it yet. See you in the next class. Don't forget to subscribe to our channels, hit the notification bell, and share the videos to your loved ones and friends that will benefit from it. Bye for now.